Any questions about anything? Okay, let's back up just a little bit and kind of re-establish a context for what we started talking about and what we're going to be talking about. So we started out yesterday talking about the importance of chromosomes and how chromosomes are these important structures within a cell that do some interesting things like encode genetic information and duplicate very precisely prior to both mitotic and meiotic cell division. So that's one of the reasons why people were interested in chromosomes. They were interested in questions of function. Well, how do you dig into understanding function? One of the ways is to understand structure. So, what are chromosomes made of? They're made of DNA and they're made of protein. So then the question becomes a little bit more focused. Which one of those components, for example, is the part of the chromosome where the genetic information is stored? Is it DNA or is it protein? We said that in the 40s up into the early 1950s, the smart money was kind of on protein because proteins were more diverse molecules. There were more monomers that made up the protein, 20 amino acids, than there were monomers that made up DNA, 4 nucleotides. And so people just logically assumed that a molecule that had to encode all this complex information for everything a cell does would probably be the more diverse molecules, protein. But that had to be tested and evaluated. So we talked about the first insight that came into this question of, is the genetic material DNA or protein? The experiments that were done by Griffith with his studies of pneumonia and the streptococcus bacteria. And as a result of his experiments, he discovered this interesting phenomenon that we call transformation. The ability of DNA from one cell, in his case a dead cell, to be taken up by a living cell. And when that living cell took up this DNA from the dead cell, it gained a new ability. It now had the genetic information that told it, here's how you make this capsule. And this capsule is what protected those bacteria, those smooth forms of bacteria, those virulent or killing bacteria, and allowed them to escape the immune system and survive inside of mice and cause pneumonia and kill them. That was an interesting observation and one we're going to take advantage of in lab, because you're going to do a simple experiment where you're going to transform an E. coli bacteria with genes from a jellyfish. And you are going to transform that bacteria so it can now do something it didn't do before. It's going to be able to glow in the dark when you shine a UV light on it. It's going to have been transformed by taking up this gene for this protein called green fluorescent protein. And we'll talk about that a little bit more next week in our Monday session. Fast forward about 16 years, and Avery McLeod and McCarty did a follow-up experiment to Griffith's transformation experiment, and they were trying to determine what was the substance that was being released from those dead, heat-killed, virulent cells that was transforming the smooth, avirulent cells into virulent ones. So they did the same experiment basically, but instead of using the entire cell, they isolated the cell that had been heat killed into its 4 main molecular components: carbohydrates, proteins, lipids, and nucleic acids. And they repeated his transformation experiment with those isolated components. And the only component that transformed the cell successfully was the nucleic acid component, specifically the DNA. So that was pretty compelling evidence that DNA was the genetic material and not protein. But it didn't lay that particular question completely to rest. It wasn't really until 1952 when another very elegant experiment was done by Hershey and Chase that really fully convinced everyone DNA is the genetic information and not protein. Hershey and Chase weren't working with bacteria and pneumonia. They were working with something very different. They were working with something called a T phage. Anybody know what a T phage is? When you see that word phage, it should say something to you. A phage is a virus, and specifically, it is a virus that infects a bacteria. Now, we're going to talk about virus later in the semester. Right now, most people are aware of viruses in the context of human disease. And I think there's this misconception that the only virus that exists out there are viruses that infect humans or other animals. But there are plant viruses. There are fungal viruses. And there are bacterial viruses as well. And these phages are viruses that infect bacteria. The T phage has a quite interesting anatomy. If you look at a T phage, it consists of this kind of bizarre looking, almost alien spacecraft looking structure. And this structure, called a capsid, has some docking sites on the bottom of it called tail fibers. So these are the tail fibers, and this portion is the capsid. And all of this structure is made out of protein. Packed up inside of the capsid in the head region is the genetic information. And the particular virus that they were using, the genetic information was DNA. You're going to see later on that not all viruses have DNA as their genetic information. Some have RNA as their genetic information. But these were DNA viruses. These particular viruses are able to infect certain types of bacterial cells, and the infection process involves the virus attaching to the surface of the bacterial cell. So there's got to be some complementarity. There's got to be docking sites on the bacteria that match up with these tail fibers or receptors on the virus, and the two have to fit together before infection will take place. That's why we're going to see later on that only certain type of virus will infect certain types of hosts. If they can't attach, they can't affect. What happens following attachment is the virus literally injects its nucleic acid into the cytoplasm of the cell. And so it's almost like a hypodermic syringe, and it plunges through the cell membrane and the cell wall of the bacteria, and the nucleic acid is pushed out of the virus and into the cell. So now we've got this kind of empty husk sitting on the outside of the cell that's made of protein, but it has pushed its nucleic acid out into the cytoplasm of the cell. So what happens when you have viral nucleic acid in the cell now? The viral nucleic acid is kind of a cell terrorist, and it takes over the cell, and it reprograms it. And it reprograms it to do one thing. What's that one thing? Does anybody know? Produce more virus. It turns the cell basically into a virus factory. So it transforms that cell. Okay? So Hershey and Chase were virologists, and they knew this particular information. And they knew that whatever got into the cytoplasm of the cell, whatever was being injected from the virus, was the genetic information of the virus. Okay? So they did a very elegant, somewhat simple experiment to try to determine what was getting into the cytoplasm of the cell. Now, unlike Avery, McLeod, and McCarty, they had a simpler system to work with. Avery, McLeod, and McCarty had to work with bacterial cells that had protein, nucleic acid, carbohydrate, and lipid. Do viruses have carbohydrates? No, typically these phages don't. Do they have lipids? No. They're made up of two components, protein and DNA. Okay? So the problem was much cleaner. It was going to be much easier to determine which of those two things were being injected into the cell. One of the tools that they used in this particular study were radioactive isotopes. And two of the radioactive isotopes that they used were a radioisotope of phosphorus, P32, and a radioactive isotope of sulfur, S35. We haven't gone over the structure of DNA yet. But do you know from past experience, does DNA contain any sulfur? It doesn't. Okay. It has nitrogen, it has phosphorus, it has carbon, oxygen, hydrogen, but DNA, no sulfur. Does protein have sulfur? Typically, okay, there are some amino acids that contain sulfur. And typically those amino acids are part of common proteins. So protein contains sulfur, DNA doesn't. How about phosphorus? Well, DNA has phosphorus. Does protein have phosphorus in it? Nope. Remember what an amino acid looks like? It's got this central carbon, it's got an amino group, it's got a carboxyl group, it's got a hydrogen, and then it's got that wild card, the R group, and none of the R groups have phosphorus as part of it. Okay? So what they did was they grew up populations of radioactively labeled virus by growing them on media that contained these isotopes of P32 and S35. So the P32 selectively labeled the DNA, and the S35 selectively labeled the protein. So there was a way to differentiate between those molecules based upon the radioactive tag that was put onto them, basically. This is also sometimes called the blender experiment, because a kitchen blender was used as part of the instrumentation in this experiment. Does anybody know what the purpose of the blender was? Look at this particular diagram up here. See if you can figure it out. Okay. What they were wanting to do is they were wanting to determine what the virus that infected these E. coli bacteria were injecting into the cytoplasm. Okay. Was it the S35 or was it the P32? Remember we said that these were kind of empty husks that were left on the outside of the cell after the injection had taken place. What do you suppose would happen to these cells and these husks if you put this into a blender and turn the blender on really high? What would it do to this interaction right here? Think they would still be attached? Probably not. Okay, the shearing force of the blender would knock these virus off of there. And then if you centrifuge that liquid, what would happen? The cells would be pelleted, and these knocked off capsids would remain in the liquid portion or the supernatant. So now all you had to do was figure out which radioactive label was in the supernatant and which one was in the bacterial cell. And which one turned out being in the bacterial cell? The P32 labeled material was in the bacterial cell in the pellet, and the S35 protein labeled stuff was in the supernatant. And basically that's what this particular experiment shows you. It gives you the two different scenarios of growing up one set of bacteria with the sulfur label on it and see what happened to that. The radioactivity stayed in the liquid following blending and centrifugation. Second experiment involving labeling the bacteria with P32 phosphorus. And here the P32 phosphorus was in the pellet, meaning that the stuff was inside the cell. So the label associated with the DNA was what was getting into the cells. Whatever was getting into the cells was the genetic information. So DNA and not protein was the genetic information. Pretty cool experiment. And very definitively then finally answered this question: DNA is the genetic information. So now people could be a little bit more focused. Because again, remember, we're trying to answer this question. The first question was, how do chromosomes replicate prior to mitosis and meiosis? How do chromosomes store genetic information? And now people are asking a more focused question. How does DNA do that? Because DNA is the real stuff. It is the genetic information. So to answer questions of function, storage of information, replication, what's a good strategy to use? What is the structure of the molecule that we want to understand the function of? Okay? So that's where Watson and Crick and others come into play. From around 1950 until 1953, this incredibly competitive research was going on in several different places around the world to try to answer this question: what is the structure of DNA? This was kind of the holy grail at the time of biology. This was the question that people knew that if they could answer, they would have long-lasting fame and notoriety and probably get a Nobel Prize for it. Here are the major players in this race. Watson and Crick, you've heard of. I mentioned Rosalind Franklin. Maurice Wilkins also worked with Franklin. We'll talk about their contributions. And then people that you probably haven't heard of, I doubt you've heard of Chargaff. Some of you may have heard of Pauling, Linus Pauling. Does that name ring a bell to anyone? We'll talk about what Pauling's contributions were as well. But let's start out with Watson and Crick. Two pretty interesting individuals. This is a picture of Watson and Crick looking at a model that they had constructed out of ring stands and tin that they had cut into pieces to make molecular models. And they would put all of these pieces together and try to develop this structure that they thought represented what DNA looked like. So that's one of the ways in which this particular research that was being done was kind of interesting. This is really an exercise in modeling. Now, as it turns out, a model is just a type of hypothesis. But it's kind of a physical hypothesis, okay? It's one that you can actually manipulate. But what do we have to do with all hypotheses to verify them? Think back to last semester. Dr. Burns goes, please, come on. So if you have a hypothesis, what do? You need to test it, right? You need to apply information that either you gather or other people have gathered to this hypothesis and say, does the information support it or does it refute it? What if the information refutes your hypothesis or is inconsistent with the way you've built your model? What do you have to do? You change the information? Hopefully not. That's not scientifically ethical to fudge your data so it fits your hypothesis. What do you have to do? You have to change your hypothesis. Sometimes It's just so out of whack, you have to discard your hypothesis and develop an entirely new one. But sometimes it requires a little bit of tinkering, a little bit of modification. And that's basically what they did, is they had to build this model based upon information that they gathered and to see, is the information consistent with their particular model. Now, where they were a little bit unique was that Watson and Crick really did very little experimentation themselves. They did very little data gathering themselves. They used other people's data. Is that okay? Well, it depends on how you do it. Okay? If the data has been published, you certainly have access to that data. I mean, anybody can read a published scientific article and use that information in their research. That's the basis of science. Where you get into trouble is when you start using information without permission. And some of that was going on during this particular process. And that's where Franklin comes in. The backgrounds of Watson and Crick are kind of interesting. Watson is an American, and he was a biologist. Crick was British, and he was a physicist. There was really no such thing at the time as a molecular biologist. Okay? That role hadn't really kind of been created yet again. So what did some of these other people contribute? Watson and Crick, as it turns out, were very good and adept at kind of taking data from many sources and putting it together and developing a model. But where did the data come from? Well, Chargaff had done a lot of work with nucleic acids, in particular DNA, and developed what later became known as Chargaff's rules. He looked at DNA from all kinds of different organisms. And basically what he determined is the amount of adenine in DNA was always equal to the amount of thymine. So if you did a determination of percentages of A, T's, G's, and C's, A and T were usually roughly equivalent to one another, as was the amount of guanine and cytosine. That turned out to be a very important realization that Watson and Crick used to develop their successful model. Another set of information came from a technique called X-ray diffraction. X-ray diffraction was, as it turned out, kind of a dangerous technique that involved bombarding crystals of molecules with X-ray beams. And as the X-ray beams passed through the crystal of the molecule, the X-ray beams were spread or diffracted as they collided with the molecule in the crystal. And there was a photographic plate behind the molecule. And so you could see a pattern of diffraction that was shown as molecules were bombarded with X-rays, and the X-rays were bent as they passed through the molecule. And you got these very interesting patterns. Here is one of the most famous ones right here. This is Rosalind Franklin's X-ray diffraction photograph of DNA. And this is a photograph that Watson and Crick got a hold of, without her permission, that was instrumental in them developing their successful model of DNA. This was a big controversy that didn't really come to light until many, many years later. As I mentioned before, Watson and Crick and Wilkins, who worked with Franklin, were all recognized for their contribution of discovering the structure of DNA by getting the Nobel Prize. Rosalind Franklin was not. Okay? There's actually a video that is, I think, still available on YouTube called The Secret of Photo 51. This is Photo 51 right here. And it talks about Franklin's life and the whole controversy that occurred as a result of her being dissed, basically, when the credit was being given for the discovery of structure of DNA. There's an optional point opportunity that's listed on the website that involves watching the video and writing a summary of the video. So if you're interested in learning more about Franklin, take a look at that and you can do that. There were some important insights that trained people like Franklin, not Watson and Crick. They didn't have the training to interpret these photos, these X-ray diffraction photos. But these are some of the insights that Franklin was able to develop about DNA, including what the diameter of the double helix was, how closely stacked together the purine and pyrimidine bases were, the amount of distance it took to make the helix that they were proposing make one twist, how many nitrogen bases there were at every turn of the helix. Some very specific physical information about the molecule was deduced from looking at this X-ray diffraction pattern. Now, when Watson and Crick saw it, they knew enough about X-ray diffraction to look at this particular pattern and say, I know what that is. This is characteristic of a molecule that is a double helix or a two-stranded, twisted molecule where the strands twist around one another. So they knew that much, and when they saw this particular photo, they went, wow, that's really important. So then they worked with her collaborator, Wilkins, to figure out the rest of this information, kind of leaving her out of the loop. One of the insights that was particularly important from this physical information obtained from X-ray diffraction was the fact that DNA molecules had a uniform diameter. They weren't lumpy, okay? The helix had a fairly constant diameter. It wasn't wider in some places and narrower in other places. Well, as it turns out, the nitrogen bases, the A's, the T's, the G's, the C's, are of two types. There are purines and there are pyrimidines. Purines are bigger molecules than pyrimidines are. So one of the things that they deduced from looking at this information was, if the molecule has a constant diameter, the only way that really can make sense is if a purine on one strand is always paired with a pyrimidine on another strand, a big one and a little one. If two little ones were paired, then the molecule would be narrower at that point. If two big ones were paired with one another, the molecule would be wider at that point. Okay? So this suggested purines being paired with pyrimidines. That was an important insight. This basically is the paper that started the whole genetic engineering phenomena and started molecular biology. One of the things that's very significant about this particular paper is it's 1 page long. That's unheard of now in a scientific paper. Okay? 1953, the molecular structure of nucleic acids. You do not see Rosalind Franklin's name on this paper. Watson, Crick, and Wilkins. This is posted on the website. I encourage you to look at it and read it. It's really interesting how much can be packed into this one little 2-column paper that changed the history of biology. What are the major insights that they reported in this 1-page paper? That DNA was a double helix, that it was 2 strands wrapped around one another. That the 2 strands are held together by hydrogen bonding between what we now call complementary base pairs. The A on one strand pairs with the T on another strand. The G on one strand pairs with the C on the other strand. Consistent with Chargaff's rules. That's why we always find equal amounts of A and T and G and C in DNA. And importantly, didn't seem so important at the time, but we'll see why it's significant now. DNA is an anti-parallel molecule. Does anybody know what that means? As it turns out, the two strands of DNA have polarity. And basically, we call one end of the molecule the head end, and one end the tail end. So if this is one strand of DNA, and this is its partner, the two strands are lined up opposite one another, or anti-parallel. And so if this one runs head to tail, this one runs tail to head. That's really going to be significant later on. Okay, here's a representation of what a single strand of DNA looks like. So we're looking at this one strand, for example, now. Sometimes people will say, okay, they'll give it a name. They'll say, this is the Watson strand, and then its complement is the Crick strand. Okay? So this is one of the two strands. So what do we see here? Well, we see that this strand of DNA, this single strand of DNA, is a polymer. And it's made up of repeating units, which we call nucleotides. So this single strand of DNA is a polynucleotide chain. This particular polynucleotide chain has a thymine hooked to an adenine, hooked to a cytosine, hooked to a guanine. But it doesn't have to be that way. This could be an A, this could be an A, this could be an A, this could be an A. There's no limitation as to which nucleotide can be hooked to another within a given strand. This particular bond right here that attaches the sugar of one nucleotide to the phosphate of an adjacent nucleotide is called a phosphodiester bond. Polynucleotide chains are a string of nucleotides joined together by phosphodiester bonds. Just like a polypeptide chain is a bunch of amino acids hooked together by what kind of bond? What hooks together amino acids in polypeptide chains? Bio-ret test you did in lab specifically identified this particular kind of bond. Peptide bond, right? Okay. Now, one thing I want you to notice here. This 4 nucleotide sequence has a beginning and an end, right? This end looks different from this end, right? Because this one, the last group, is a phosphate group. Down here, the last group is a sugar, specifically the sugar deoxyribose. That's what DNA stands for, deoxyribonucleic acid. Well, we call this end of the molecule, if you want the head end, right here, the 5' end. And we call this end down here the 3' end. Get used to that designation, 5' and 3', because it's going to be important when we talk about DNA replication. It's going to be important when we talk about transcription of DNA into RNA. You got to know what that means. You've got to appreciate that. Well, why do they call it 5' and 3'? It really has to do with the sugar and the carbons in the sugar. See these numbers right here? Deoxyribose sugar has 5 carbons. There's a carbon, there's one, there's one, there's one, and there's one. They're all labeled. Okay? So the phosphate group at the head end is attached to the number 5 carbon of the sugar. That's why we call it the 5' end. Down here, there's no phosphate group because this is the end of the molecule attached to this 3 carbon. Instead, there's a hydroxyl group down here on the number 3 carbon. That's the 3' end. So you see how the two ends, the 5' and the 3' end, are different from one another? They terminate in different functional groups. Phosphate group here, hydroxyl group over here. Well, that's one strand. The DNA is a double helix. The key to holding the two strands together is hydrogen bonding between complementary nucleotides on one strand and the other strand. So now we're talking about interactions between the Watson strand and the Crick strand. And now there are some limitations as to what can be across from one another in the molecule. This is where the base pairing rules come into effect. Adenine can only be across from thymine. And when they're across from one another, they form hydrogen bonds. How many? There's a hydrogen bond here, and there's a hydrogen bond here. And so that's when we represent the base pairing between adenine and thymine. We have two little bonds shown here representing the two hydrogen bonds. Here is the base pairing between guanine and cytosine. So this is on one strand, this is directly across from it on the other strand. Guanine and cytosine form 3 hydrogen bonds with one another. Okay. So which one of those AT or GC base pairs are going to be held together a little bit more strongly? Guanine and cytosine. That extra hydrogen bond gives you some extra strength. So, here's what a DNA two-stranded molecule looks like, but not shown in the helical representation. This is just to make it easier. So here's one strand, 5' end, 3' end. Here are the nucleotides in that particular strand. No restriction as to what the order of these nucleotides can be. And then here's the complementary strand. Now there's restrictions. Because if this is T, this has got to be an A. That's the only ones that fit together properly. If this is a G, this is a C, etc. And notice the anti-parallel nature of the two strands. Head, tail, tail, head. Now to get a DNA molecule, all you do is you just twist this up. And this shows you the twisted nature of the DNA double helix. The blue right here represents what we call the backbone of the molecule. What does the backbone consist of? Sugar, phosphate, sugar, phosphodiester bonds joining those nucleotides together. So that's what these blue stripes indicate right here is that backbone. And then attached to them are the nucleotides which have the complementary base pairing. A across from T, G across from C. And this shows you some of the physical parameters of the double helix, which are consistent with the X-ray diffraction data that they obtained from bombarding DNA molecules with X-rays. So the diameter of the molecule, the distance from one turn of the helix, the distance between consecutive nucleotides on one strand. This all fit together. The physical information was consistent with the model that they developed. It was a great discovery. But where did that get us? Okay, this gives us information about the structure of DNA. But this whole quest was really grounded in this idea of we want to understand function of DNA. How does it store genetic information? How does it replicate? Well, can you get any insights into those questions of function just by looking at this structure? Well, not so much the question of how it stores genetic information. That's pretty complicated. But once Watson and Crick were convinced that this indeed was the proper structure of DNA, they had a real eureka moment. And we'll talk about that in just a second. One little aside. There's one name I haven't mentioned yet. Linus Pauling. Linus Pauling, if you've ever heard of him before, unless you're a Nobel Prize groupie and understand that he won a Nobel Prize for protein biochemistry early in his career, you might have heard of him because for a long time, Pauling was a big proponent of vitamin C being a potential cure for cancers, a preventative for colds, a preventative for flus, and he was this big vitamin C guru, and he advocated taking these massive doses of vitamin C. And a lot of people listened to him because he was a former Nobel Prize winner. He was a biochemist. As it turns out, that particular idea really didn't gain much traction, and while vitamin C has its benefits, it doesn't appear to work the way Pauling thought it did. But he was involved in this race to discover the structure of DNA. Watson and Crick were working in a lab in England, nearby where Franklin and Wilkins were working. Pauling was working at Stanford?
I think it was at Caltech.
Caltech? Okay. In California. And so these two groups were racing with one another to discover this structure. Watson and Crick felt like kind of the poor stepchildren because they didn't have the notoriety Pauling did. And they never really believed that they were going to beat Pauling. One of them, I forget which one, it might have been Watson, knew a relative of Crick's, a nephew, maybe even a son, and got wind of the fact that Pauling had discovered the structure of DNA. And they were just, oh man, we've been beat. And they got a pre-publication, a preprint of an article he was getting ready to publish. And that's acceptable, you can do that. And they started madly looking through this article, and they found out that Pauling was proposing a structure which was a triple helix structure of DNA. And they started looking at it and they went, wow, this really doesn't make sense. I think he screwed up. Particularly because they had access to the X-ray diffraction data. And Pauling didn't have this photo 51 that was so critical. So they started reading through it and they went, man, he's made a mistake. This can't be right. And as it turns out, it wasn't. And so they got some new life. They continued on with their research and eventually were successful and were the Nobel recipients. What was their eureka moment when they looked at this particular structure? At the end of their paper, they stated something which seemed like very innocuous at the time, but that was very significant. What they said at the end of our paper is that it has not escaped our notice that the specific pairing that we have postulated immediately suggests a possible copying mechanism for the genetic information. So by looking at that structure, they went, I know how that copies. I can now answer a question of function by knowing the correct structure of that particular molecule. Their belief was that DNA replication occurred in a manner that they referred to as being semi-conservative. What does semi-conservative replication look like? So, if here's a DNA molecule, let's say this is the Watson strand, this is the Crick strand. Everything's kosher with this molecule because the A's are pairing with the T's and the G's are pairing with the C's. If this molecule were to replicate prior to mitosis and meiosis, what do we need to have happen? We need to have this parent DNA molecule give two daughter DNA molecules, or chromosomes, that have the identical sequences of A's, T's, G's, and C's as the parent does. How can you do that? Well, if the two strands separate from one another, what does that involve? That involves breaking hydrogen bonds. That can be done, particularly if an enzyme is involved that can catalyze that reaction. Then each of these strands can serve as a copy surface for the formation of a newly synthesized strand. That idea of a copy surface, or what we call a template, was not a new idea. That was used in manufacturing all the time. To take something and make a copy of it, to have a template and then make something new out of that template. So their belief was the DNA molecule separated into its two strands, and then this one was used to make a new strand, and this one was used to make a new strand. So if we represent the DNA molecules in this fashion, the solid lines represent original or parent strands of DNA. So what happens? The two strands separate. Okay? And then what do? We then synthesize from this strand a copy. And we synthesize from this strand a copy. So this is now one of the daughters, and this is the other daughter molecule. They have the same sequence of A's, T's, G's, and C's as the parent did. But each strand consists of part of a parent molecule and a newly made one. Part of the parent one and a newly made one. Some of the parent was conserved in both daughter molecules. But not all of the information was parent information, so they called this semi-conservative replication. So here's our original parent. Here's separation into the two strands. And then this strand is serving as a template for making a new strand. This strand is serving as a template to make a new strand. How does this new strand know what to put here? A's got to pair with T. G's got to pair with C. So whatever the sequence in this strand is, dictates what the sequence in this strand is. Sequence in this strand dictates this one. But look at the relationship. Parent, AT. Daughter, AT. Daughter, AT. CG. TA. The sequence of this one is identical to this daughter and this daughter as well. So this just shows you the double helix then unwinding and the original parent strands serving as templates to make newly synthesized strands. Here's something important for next time. Is there any experimental evidence for semi-conservative replication? It makes sense that this is how it happens. But just because something's logical doesn't mean that's the way it happens. Remember when we talked about photosynthesis? We said it seemed logical that carbon dioxide was split. But what turned out to be the case? Water was actually being split. So we'll talk next time about how this semi-conservative replication was analyzed and evaluated using experimentation.